Okay, good afternoon, everyone. This is our lecture 13. Today, we're going to finish our discussion on the dimension reduction method. So as a recap, we have discovered the purpose of doing dimension reduction, including data visualization, and to reduce the dimension to get around the curse of dimensionality, and to remove noise by keeping only the major dimensions, which preserve some important data characteristics. Okay, so we call the low-dimensional data after the reduction as low-dimensional embeddings. The first and classical method we introduced is the PCA. So for PCA, everybody is most familiar with this. So what it does is try to find some low dimensions to preserve the variance in the original data. And the nice property is that you are going to have the PCs orthogonal to each other. And each new dimension is corresponding to some linear combination of the original P dimensions. And we call those P weights in each new dimension as loadings. So one important thing to note is about the use of PCA in practice. That is, when you apply PCA, would you want to standardize your data or not before you run PCA? By standardizing, I mean, do we want to make the column of my data? So here, each column is for one feature, each row is for one data point. Do we want to make each column to have standard deviation equal to 1? So to answer that question, that's a commonly faced question. We need to really think about the unit in our data. Do we trust the units of the original features, want to preserve the units, or we don't care about the units? So if you don't care about the units and you want to make every feature on the same ground, then you want to do standardization. And this is common in social science. Like if one column corresponds to ages, the other column corresponds to income, they clearly do not have the same unit. You don't want to have the feature that has a larger magnitude nominally to dominate the PCA. In that case, you want to do standardization. Then for our gene expression data, the units are comparable, like they're all RPKMs or TPMs. So in this case, you do not want to do standardization. So yes, it is case by case. And another thing to keep in mind is that to really understand PCA, I think the key is to understand the SVD. And SVD and Eigen Decomposition for the X transpose X. So we can either do SVD on data X or Eigen Decomposition on X transpose X. I think understand those can make you really understand PCA to the ground. But if not, you can still think of PCA as a way to preserve variance, and the PC1 preserves the most variance, PC2 less and less and less. And you want to look at the cumulative variance preserved by the top K PC to help you decide where to set K. Okay, that's PCA. And then we talk about the Pnmf. So Pnmf is something which is similar to PCA, but it's similar to PCA in the sense that you are also finding a linear combination of the original features into the new features. But the difference is that Pnmf requires the loading matrix and the final embedding matrix to be both non-negative. This is something PCA doesn't have. So this non-negativity will have some advantages, including sparsity. So sparsity means that in the loading matrix, each new dimension only has non-zero loadings for a small number of original features. So that can make interpretation very good. And as a generalization or extension, we have the Pnmf, projection Pnmf. This addresses the issue that Pnmf does not provide a projection matrix as PCA does for projecting new data. So Pnmf, in a sense, in other words, is more similar to PCA by having the projection property, and it is also similar to Pnmf by having the non-negativeness. And also because of this objective function, so you see that we want to have this W transpose W close to the identity matrix so that we can have this loss function very small, right? That's our goal. And so driven by this goal, then this W matrix will have its rows roughly orthogonal to each other. Okay, and the W transpose, as we said last time, it is a projection matrix. So then what does this mean? So here it has P rows and K columns. So we can think of each column as a loading of each new dimension. Okay, to put it that way. And the fact that we want to have this W transpose W close to identity, although we know it's not, so this can actually imply, okay, if I do it in the other way, W times W transpose, then I would have a identity matrix close to be a K-dimensional matrix close to the identity. So what that implies is that I really want to have the K new dimensions roughly orthogonal to each other. So the sparsity, non-negativity plus the orthogonality will give me what? I would have these K rows to be roughly non-exclusive. So whenever I have non-zero loadings in one new dimension, these features are likely to have zero loadings in all the other new dimensions. So this makes each feature, each new feature, correspond to a unique set of original features that can also enhance the interpretability. So I would say compared to PCA, Pnmf has two clear advantages: interpretability and also its non-negativity. That may make more sense for you for your new data. But it definitely has drawbacks. One is that it's computationally slower. And second, it may not give you a unique solution if you try different initialization. That's also the case for NMF. Well, for PCA, we don't have this issue. We always get the same set of PCs. And also, it doesn't preserve the variance as well as PCA does because it doesn't have that objective. So this can also make you realize that we cannot have an algorithm that achieves all the benefits. There must be some trade-off depending on what you look for. Okay, so PCA and NMF were introduced together. Then we introduced the MDS, multi-dimensional scaling. So different from PCA or NMF, for MDS, we don't need to start with our data matrix X. We just need to have a pairwise distance matrix, n by n matrix. So we have the endpoints and we have their pairwise distances. So this is a symmetric matrix. And we just start from here. And our goal is to find a two-dimensional or three-dimensional embedding of n data points so that their pairwise Euclidean distances in the two-dimensional or three-dimensional space is as close to the D as possible. So that's our goal for MDS. And if we know that the MDS distance matrix was calculated as the Euclidean distances from the original data, when that happens, then what classical MDS, which is what we introduced, is equivalent to PCA. Okay, but it is not required that Dij must be Euclidean distances in the high dimension. So that's why MDS can be considered as more general than PCA, and it requires less for the input. Okay, then at the end of our last lecture, we introduced tSNE. So the beginning of tSNE, and its original form is called SNE, Stochastic Neighbor Embedding. So by the name, you can see that it thinks about neighboring data points, and it is a stochastic algorithm. So what it does, so you have two sets of data points, x1 to xn in high dimensions, y1 to yn in low dimensions for your plotting. So your goal is to preserve the pairwise relationship in the high dimensions by this pairwise probability. So which means that unlike the Dij in MDS, which is just a distance, here you're not taking a distance and you're not treating I and j symmetrically. What you did is that for each I, you consider it to be the center, and for its neighbor j or every other data point j, you calculate the probability for I to pick j as a neighbor. And this probability is proportional to this density, or you can call it kernel transformation of the high dimensional distance. So this is the Euclidean distance in high dimension. What you did is that you put this distance into a normal density. This normal density has mean 0 and variance sigma I squared. This sigma I is going to be determined by data point I as the center. But anyway, after the normalization, you are going to assign each j not equal to I a probability of being chosen as the neighbor, so that they can be taken the sum to 1. So what I forgot to say is that for this, we make a special case. So this is for the case where j doesn't equal to I, okay? And we also have the special case for j equal to I. In this case, we just set it to 0, okay? And for the low dimension, we do the same thing. For j doesn't equal to I, we assign the probability proportional to this density transformation. And here we know that we arbitrarily set the variance for all data points equal to 1/2. So we don't need to worry about that variance term anymore, unlike in the high dimension. This is reasonable because the high dimensional data are observed. So therefore, we cannot really control the spreadness of the data. So we actually use the sigma I squared to account for the fact that for some data points, its neighbors may be close to itself. So we have a very tight cloud. For other points, we have their neighbors far away from themselves. So then we want to take that into account. So in other words, the sigma I squared is changing locally for different centers. This is the fact why this tSNE or the SNE can capture the local clustering structure in our data. And this is why it was popular. It has been popular in single cell data analysis, because for single cell data visualization, our primary goal is to visualize the cells so we can see cell types or cell clusters. And we do not really care whether the cell cluster sizes in our two-dimensional visualization are realistic or they are correlated with the actual cell cluster sizes in high dimension. We give that up. So that's why tSNE is advantageous in that. But for PCA, you can imagine it captures the variance in the data overall. That means if you have a cell cluster that's really big and really large in the high dimension, by large, I do not mean that it has a lot of data points or cells. I mean that it's really spread out. When that happens, then the PCA will be dominated by that large cluster. Then you're going to miss the small clusters in your visualization. So those cannot be really captured by plotting just PC1 and PC2. But tSNE, since this adaptiveness of its local density, it can make sure that for each point, we can collect its neighbors, no matter being far away or close, depending on the density around I. And then in the low dimension, I make every point I have the same spread out, same variance. So I can equally present those clusters. Okay, for this low dimension QII, for I picking up itself as neighbor, we set it to 0. So then the leftover is to find the Y1 to Yn, which are in the low dimensions, so we can make QJIs close to PJIs as much as possible. Okay, so in this case, the original SNE paper basically uses this so-called callback Leibler divergence, short as KL divergence, to find QJI. Because we can think of, we said last time, okay? So QJ given I give us n distributions. We have one distribution for each I as the center. And this distribution has how many possible values? It's actually n possible values. It's the n points. For each I, what is the probability for it to pick any of the points as the neighbor? So that itself is an n-value discrete distribution. And we have an n-value discrete distribution for QJI for each I. So we want to make them close for each I. And we have how many of those distributions? N, because I can go from 1 to N. So together, we are going to make them closer. So what I mean, to put it in words, what I mean is that we define distribution. We call them Pi. So Pi would actually be the collection of Pj given I, okay? And we let J go from 1 to N. So this is an N value distribution. These Pji's, if I take the sum, equal to 1. I also define it one for the low dimension Qi. So Qj given I and J from 1 to N. So then I can talk about the KL divergence. KL divergence of, so usually for KL divergence, we need a reference distribution as the baseline and a distribution we want to evaluate for how close it is to the reference distribution. Here, the reference distribution is clearly the high dimensional one, the one we observed. So we put Pi as the reference distribution, and the distribution we want to evaluate, we call Qi. Okay, and then by definition, it is this. For discrete distribution, here's the definition. We go through all the categories to evaluate probability. J go from 1 to N. We have the reference probability Pj given I times the log of the reference probability Pj given I divided by Qj given I. That's the KL divergence. And we want to find the Qj given I to be as small as possible. But this is just, and you know that KL divergence is a very widely used thing in machine learning for to make two distributions similar in the training. So what we mean is that we want to minimize the KL divergence to find the Q. But please bear in mind that it's not a symmetric distance between two distributions because Pi is used as the baseline. And another thing we want to know is something we call the Wasserstein distance, which is also used in machine learning for measuring the distance between two distributions. And it has very important applications in the neural network. If you want to train a neural network to generate data that looks like real data, then Wasserstein distance is used for the training. But we are not going into detail for that. Okay, this is just for the ith point as the center. But together, we will need to add up for I. So then the loss, we call it C, is defined as the summation of the KL divergence between Pi and Qi. Yeah. Oh, and by the way, I can say that you can see that why the KL divergence will encourage the Qji to be equal to Pji, because you can see that when Qji equal to Pji, we have log of 1, and that's 0. So then the KL divergence is going down to 0. And we can show that mathematically, the KL divergence is non-negative. So the smallest possible KL divergence is equal to 0, and that happens when all the Qji's equal to Pji's for J from 1 to N. So that's something good to know. So then putting them together, we have this loss function, and we want to minimize it. And our goal is to find y1 to yn. And we know that y1 to yn are used in Qji by this relationship. So that's why this objective function, find y1 to yn by minimizing C, okay? And clearly you can see that C itself is a function of y1 to yn. So in other words, we look at C as an n-dimensional function. So its input, sorry, not n-dimensional, that's not correct, let's say it. Its input is y1 to yn, each is 2-dimensional vector. And what we get out of it, the C is a scalar value, that's non-negative. So this becomes an optimization problem. And in optimization, it's usually to deal with the maximization problem or minimization of a function. So a very famous approach some of you might have heard of is what we call the gradient descent algorithm. And that's what's implemented here and also in many places for machine learning. So what does that mean? Here we'll just briefly say it. Gradient descent algorithm. So let's use a two-dimensional case as a way to indicate what it means. Okay. So basically, what it does is this. So let's say that we have a function that's just one-dimensional. Probably that's easier to understand, x and fx. So if the function has something like that, and it has multiple local minimum, okay, so it depends on where we start. We will need to find the local minimum. That's the best we can do. So of course, the approach everyone is familiar with is we can take the derivative of fx and set it to 0 and find which values of x makes the derivative of f equal to 0. And these x's are the solutions corresponding to the local minimum. That's the thing we're familiar with. But what if that is not achievable? So when you cannot have prime x, this is difficult to solve, okay? So this easy solution is no longer available. What can you do? The gradient descent is a numerical approach to help you achieve that. So what it basically does is this. So you start from somewhere, let's say x0. You evaluate the derivative. Okay, you evaluate the derivative at here. So that would be fx0. So the derivative, we know that in the one-dimensional calculus would be the slope of the line tangent to the curve. That would be our derivative. So you know that if we think of the magnitude of the sign, so clearly the sign is negative here. And what we want to say is that if you want to think of this magnitude, then the magnitude reflects the steepness of the line at this point. So you can think of this as how much you want to go and what direction you want to go. If you want to maximize a function or minimize this function. So you know that this negative implies if you want to maximize the function, you want to go left. If you want to minimize the function, you want to go right. So that's why the gradient here is very informative about where you want to go. So therefore, the gradient descent algorithm is basically to update your value of next value. So here, the value would be our y's. Okay, so for example, if I put it as, so if I put it as y1 to yn as my argument, that corresponds to my x here. So I put this, the set, I call them a set as y. So then the gradient of the part of the c, then the gradient, I can write this as c dy, okay, at the current value of y, and I can give it a super index for the iteration, say in the iteration t. So this is actually what? It's actually equal to, the gradient becomes a set itself as well. It's partial c, partial y1t. For the first data point, I want to know how much I should go or what changes I should have given its current value, to partial c, partial y, and t. Okay, so that's the gradient. And then for gradient descent, okay, we're not doing gradient ascent. So gradient descent means that you are going to move in the direction of the gradient. So then what does that mean? So we start from our yt, but we want to update y to be yt plus 1 to update our arguments in this function. So then what we do is that we take yt and we go a step in the gradient. So that step is usually denoted by eta, and then we do this. So you see, what does this mean? It's again, we're doing an update for each element in yt. That's each data point. So if I write it out, it's y1t plus eta partial c partial y1t. So I go step eta following the gradient. And also it depends on the magnitude of the gradient. The larger the magnitude, the more I go. And you can see why. Because if you're in a place where it's steep, you know you can go further. But if you're in a place where it's quite flat, that means you have a very small magnitude of the gradient. You want to go a small step so you do not miss the local minimum. So you see that intuition here. N2 y n t plus eta partial c partial y n t. So each thing here is two-dimensional. This is also a two-dimensional. So it's a gradient itself, but it's only for y1. And then we add them up. So that's how we do the update by gradient descent. And then gradually we want to see which values of y1 to ym can minimize this loss function. That's the purpose.
Professor, sorry, I have a quick question. If it's plus eta dc dyt, like aren't you maximizing in that case?
Let me think about that. Yeah, I think this is the point I was wondering myself. I think it's really depending on how you draw the function and how you, I think plus is actually going up. Yes, because we are actually going, so I think this should be minus. Thank you for pointing that out. Yeah, thank you. And this is also given that we set eta to be positive constant. Yeah. Okay, so, and also in practice, which is something we had in the notes, that is, you may not want to go too fast. So people can do some small modifications to the gradient descent by saying that, okay, you want to go back a little bit depending on the difference between yt, your current value, and your previous value, yt minus 1. That means if you just go too fast, you may want to follow that trend. If you go slowly, you may want to keep that trend. So it's something like momentum. You want to keep that speed. Yeah, but I'm going to leave out that technical detail in the SNE paper here because it's not that important. It's for practical reasons. But I want to get the essence about this optimization in gradient descent. And in my other class, we talk about the Newton-Raphson algorithm. So in the Newton-Raphson algorithm, what's different from here is that in gradient descent, you only need the first order differentiation, the gradient. But in Newton-Raphson, you incorporated the second order derivative. So it's kind of like a correction. And that is very important because that can give you the second order derivative, unlike the first order, that's like the slope. The second order derivative gives you the curvature of the curve right there. So if you know that information, it's always nice to use it as in Newton-Raphson. So that's a more accurate optimization algorithm. And that actually second order derivative directly tell you the step size, the eta here. So you don't need to arbitrarily set it. But this is more flexible in that for some cases, like here, it may be too difficult to derive the second order derivative. So that's why we stay at the first order derivative and we just treat eta as a tuning parameter for our optimization. And this can already let you be aware that the SNE algorithm is stochastic. It depends on many factors so that your y1 and ym could be different when you run it with different random seed. And that's indeed the case for tSNE. Like different people run tSNE if they do not coordinate with the random seed setup, or even though they set a random seed, but they have different machines, different operating systems with different random seed generator, random number generator, you may not always get the same visualization. That's a known drawback of tSNE. So that's why in practice, when we use tSNE for visualizing, say, single cell data, we treat it mostly like a visualization tool instead of relying on it for downstream analysis, just like Pca. In Pca, sometimes we just use PCs instead of the original genes, the 20,000 genes for our downstream analysis. But we wouldn't do that for tSNE data, just as the Y12IN, they are stochastic. Okay. So then we have a question that's unsolved. That's the sigma I. How do we pick this one? So in the SNE paper, it introduced something as perplexity. I'll introduce that. So how to choose sigma I for each data point I. So intuitively, what it does is this. So if you have data like this, and you have data like this, okay? So for each point here, and for each point here, you're going to pick a sigma I that's large, and the sigma I that's small to capture its neighbors. So that's roughly the case. And how do we choose it? So here it defines some quantity as perplexity. And the perplexity for the distribution Pi we have here is defined as 2 to the entropy, which we use notation H, the entropy of Pi. And we have a standard definition of entropy for a distribution. So where this HPI, for discrete distribution, its entropy is defined as the summation I from 1, j from 1 to n, has n values, negative Pj given I log 2 of Pj given I. That's the entropy definition. So the perplexity is defined as 2 to the power of entropy. So you can see the larger the entropy, the larger the perplexity. And also here, because the entropy depends on our Pj given I, which depends on the sigma I squared. So to really understand that, we can compare what the Gaussian density of two sigma i's are like. So if I draw a density like this, and if I draw another density like this, so clearly you can see that the red one, and they both center at 0. The red one has sigma I small, while the purple one has sigma I large. Okay, and then if I input the same distance, xi minus xj, you can see for the same xi minus xj, the purple one will give me a larger Pj given I than the red one, right? So the Pj given I will be here and here after normalization, of course, but still you can see that as my sigma I increases, then I would have, so I can say this. So as my sigma I increases, it goes up. Then for large xi minus xj, if the distance is large, then Pj given I increases. Okay, so that's the case when what I saw here. But if I move my xi to xj to be close to 0, you will actually see that the red curve will give you a larger Pj given I. So Pj given I will decrease as you increase sigma I. So in other words, a large sigma I will make the neighbors, the close neighbors or the distant neighbors less distinguishable, while a small sigma I will make them more distinguishable. So that's the purpose of the sigma I. And then how do we use this entropy to control it? So we know the entropy implies the messiness, right? How spread out those n values are. So the largest entropy is achieved when all the pj given I, those n values are all equal. That's when you have the entropy, the maximum. That's actually equal to log 2 of n. But if you have one of the pji equal to 1, and all the remaining pji's equal to 0, you have 0 entropy. So in other words, this perplexity is reflecting how different your PJIs, the n PJIs are. If they are very equal, you have a large perplexity. If they are dominated by one single PJI with 1, others are 0, that will correspond to the case when sigma I is very, very small. So you have only one neighbor, the closest neighbor, getting a positive probability 1, others are 0. Then you will have a small perplexity. So therefore, you can see the relationship between sigma I and perplexity. So I can restate that if you increase sigma I, all the neighbors become more equal because the curve is getting flatter and flatter. You have an increase in perplexity. But if you have the perplexity, but you have sigma I very small, you have a large spike. So only a very small number of neighbors are having positive probability, others are 0. So in short, sigma I increases, perplexity of Pi increases. Okay, so that's the rationale. And then in the current tSNE package, what it typically does is trying to find, you set a perplexity. Let's say you set perplexity equal to 10. And based on this calculation, you find each sigma I such that the perplexity of Pi for each Pi is less or equal than 10. Yeah, find the largest, find each sigma I to be the largest. So yeah, so you can see that, you can imagine that we must need some efficient computation to achieve this for finding the sigma I in the high dimension. So with this sigma 1 to sigma n for the n data points, you could calculate PJI, and then you have the basis in your loss function. And then you want to do the optimization by finding y1 to yn so that you can minimize the loss. So that's the whole story of SNE. And we haven't talked about tSNE yet, but this is the idea of SNE. So you can see why when you have more data points, the computation increases. First of all, for each data point, you need to search for sigma I. And this is done just for once. And then for the optimization, because the summation is also taken for KL divergence for the I data points when it's used as the center for the distribution. So you also have that increase with the sample size n. And of course, it's not just linear in n, it's actually square in n, because you have to evaluate for each point j in relation to I. So you have that pairwise relationship to evaluate. So together, you can imagine this is a computationally difficult question. And a lot of technical challenge is in the optimization. How can we make the optimization more efficient? So tSNE was proposed as an improved version to address these issues. So the first improvement in tSNE is that we changed the definition of Pi and Qi by making the Pj given I and Pi given j symmetric. So that's a very big change in the definition. And so basically what we did is we do this. So the first change is symmetrize. So what we do is that we define just Pi given j is the equal to Pi given j plus Pj given I And we want to make sure that the summation of Pij for I go from 1 to n, j go from 1 to n. So it's n square pairs. We won't make them sum up to 1. So we want to divide this by 2n. We need an n here to make this normalized. So in other words, we're not treating each point as a center as in SNE. Instead, we are just looking encoding pairwise distances into the symmetric probability. That's the first change. And the second change is that for the low dimensional embedding, we do not use this normal distribution for the low dimensional density. Instead, we use the t distribution. Okay, so change this normal 0, 1/2 to t distribution. And I think this is just the t, I think it's T1 or T2 distribution. Yeah, so I'm just leaving that here, but it's the student T distribution. So what's different between a T distribution and, oh yeah, it's T1, correct. What's different between the T distribution and the standard and the normal distribution is in its heavy tail. So we know that the T distribution is more heavy tail than the normal distribution. So that means here's 0. And if we want to go far away from 0, you still have some reasonable probability under the T1, but you have very close to 0 probability under the normal. So we know that for the normal distribution, standard normal distribution, 95% of probability are within the 3 magnitude. 3 within 3, or 99% are within 3, 95 are within 2 from 0. So it's very concentrated on 0 compared to T. So what is the advantage of that? Why choosing T1 instead of normal? It's actually for visualization convenience. So, oh, I should finish the symmetrized part first. So this convenience here is computational. So what we want is that using this version, you can have an easier, simpler gradient for the optimization. So this is purely computational reason. And this changes for visualization reason. So we want to make the data points more spread out. I mean, by having the local neighbors more concentrated, far away neighbors more spread out in the low dimensional or 2-dimensional visualization. That's the purpose of using T1. So that's the same issue as I said here. So you see that for this means, okay, so this means that enclosed neighbors, you see, if I want to, or probably I think it's a bad drawing. So what I mean is this, no, just forget about this. I need to redraw this. So what I mean is that, so if you think about the heavy tail as T1 and the sharp one as normal, okay, here's 0. So what I mean is that if I start with a large Qij, so because I want the Qij to be similar to Pij, right? Pij is from high dimensions, Qij is from the low dimension embedding. So if I want to preserve a large Pij or Qij, I start from here. And you can see that in this case, I would do what? So okay, probably this is not a good thing here. Yeah, I have little spaces. So I have a large Qij here. So you can see that I would reach a corresponding distance here. This is the distance I would have for Yi minus Yj for encoding this density using the T distribution. But this will be the distance for Yi minus Yj using the normal distribution. So in other words, you see, for the T distribution for very large Qij, the resulting distance in my 2-dimensional embedding will be smaller. Yi and Yj are closer to preserve that Qij. That's for close neighbors. But for far away neighbors, so if I have the density down here, and I draw the line again. So you can see in this case, Ym and Yj is here, Y minus Yj, and the distance is here. So which means that for far away, so for the small density, to preserve it, using the normal distance, normal density, I'm going to have a small distance, but using the t distribution, I'm going to have a large distance. So this tells you that t distribution more distinguishes the small or the close neighbors from the distant neighbors, or proximal neighbors from distant neighbors. So that's why it give us a better visualization. So I think that's the primary reason of choosing t over normal for the low dimensional part. Yeah, and also as a result of a t, the KL divergence for the loss function is also taking a different form for the C. And that can actually also make the optimization simpler. Okay, so let me just summarize what I said. So compared to the normal distribution, probably I just put it here in T1. Okay, so for, I can say when xi minus xj small. So by saying small, I mean that in the high dimension, xi and xj are close to each other. Then using the normal, if I think of the yi minus yj and yi minus yj, under a normal distribution, I would have it bigger than. Okay, so for using T1, I'm going to get a smaller distance in the low dimension than using the normal. But when xi minus xj is large, then the relative order is reversed. So using T1, I'm going to get a larger distance. So you can see this in effect will make us more distinguish this small xij from large xij because it shrinks the small ones further, it enlarges the largest one further. So you see that better separation. Okay, so and then the name tSNE, the T comes from T distribution. Okay, do you have any questions for this part? Okay, so that's the tSNE. And another popular method for single cell data visualization nowadays, it's called UMAP. It's a newer method for visualizing. It's also doing nonlinear dimension reduction. Yeah, tSNE is nonlinear dimension reduction because you see each of our embedding, so each new dimension, dimension 1 or dimension 2 for the embedding does not correspond to a direct linear combination of the original P dimension. That's the case for UMAP. And UMAP is a lot more complicated in its methodology compared to tSNE. So it requires a lot of topology, and also some people call it the categorical analysis. So it's a branch of discrete mathematics. So if you're interested in checking out the UMAP paper, you can see what I'm talking about. So it's not really comprehensible, that paper itself, unless you have that knowledge. So that's why we're not going to talk about it. But I can just tell you some brief idea behind this topic called topological data analysis. And if you're interested, there's a new book about this topic. It's actually becoming more and more popular in biological data analysis. And you can check out that book. I actually purchased a copy myself. But yeah, I have to admit that I haven't thoroughly read it yet. So this topic, I think it's something quite cutting edge. And it's about analyzing high dimensional data, not by preserving the variance for sure, but by preserving the topological structure. So what do I mean? So to really understand that, you need to think about some distance that's not Euclidean, but something based on the shape of the data. So I can just give you a very simple way to interpret. There's an important concept called the Geo Basic Distance. Okay, So this depends on the shape of the data. So let me make that up. Just saying, okay, this is the dimension 1. This is dimension 2. And my data points has this special shape, okay? So by geodesic distance, the intuition is that we want to measure 2 points based on how far away they are each other following the shape, like here. And we're not really interested in their Euclidean distance. So to make it in a more extreme case, let's consider this U shape, okay? So in this U shape, if you look at their Euclidean distance, that's actually very close. But if you think about their geodesic distance, you have to go through this whole shape, and they are they're far away from each other. In fact, if you consider all the points in the U, they are farthest from each other. But in Euclidean distance, they're not. So this is just a rough introduction of what the geodesic distance is doing compared to Euclidean distance. So UMAP is some idea of preserving the geodesic distance in the high dimension, but we want to use the Euclidean distance in the low dimension. So that means for 2 points that are far away from each other in geodesic distance in high dimension, we want them to have large Euclidean distance in the low dimension. That's the whole idea. And so in practice, how do we define geodesic distance? It requires a way of defining neighborhoods. So what it means is that, so you can actually think of it in a very interesting way. So for each point, if I give a small size epsilon, I can define an epsilon bow around each point, okay? So you can see I can do this continuously. And then what I do is that I would connect all the points within each epsilon bow. So if 2 points are in one bow, I connect it. So then you can see imagine what happens would be something like this. Okay, I can connect it. And in some cases, I may connect 2 things at the same time, and then I do connection. So this will give you a network structure. So every point is connected to other points in the same bow, but it's possible that one point might be totally isolated if there's no other point in its epsilon bow. Then the geodesic distance can be defined as the shortest path that connects 2 points. Okay, so that can help you capture that local structure. But of course, this depends on the neighborhood size. If you change epsilon, you will change the geodesic distance. For example, you see if my epsilon both sides are like this and continue, then these 2 points will have the longest geodesic distance. But if my epsilon bow is super large, I can include those, then their geodesic distance becomes just their Euclidean distance, right? It's very small. So that's why in UMAP, just like in tSNE, all those nonlinear dimension reduction methods require some tuning parameters that needs to be set. It's not free. It's not like you can get the same result without specifying anything. But many people, when they use it, they actually didn't pay much attention to the effect of tuning parameter. If you're interested, please check it out. I think there are many nice blogs that discuss the distinction and between UMAP and tSNE. And some people are promoting UMAP for its, it says that, okay, tSNE is going to be good at preserving the local structure, but tSNE finds a better balance or trade-off between the local structure and the global structure. And Pca is clearly preserving the global structure. So they try to use the argument to defend UMAP. And I do see that they have some empirical evidence from data, but I'm not still, myself is not totally sure how to interpret this in a more rigorous way. So what's actually happening there? So I feel like this is still an open question for those computational people. Yeah. So that's
sorry to interrupt. What did you say there was a book that you'd recommend?
I wouldn't say. So can you show up when you speak up so that can help me see your face? Because otherwise, I feel like all of a sudden there's some voice coming out.
Yeah, sorry, every time I switch the camera on, like the audio gets a little worse.
Oh, okay, thank you. Yeah, no problem. So Yeah, so I just say that there's a new book, Topological Data Analysis for Biologists, some title like that. So if you're interested in checking out, you can do. But it's not something that you can just read in 1 or 2 days. It's actually a very dense book. You need to spend time reading about it. But this is the newest thing I found myself. Yeah, because I think for my own research, my students and I are really need something like this. So to really understand something like UMAP. Yeah. Okay, thank you for your question. And so yeah, that's tSNE, and that's and UMAP. And I said that you can also Google tSNE versus UMAP. There are many interesting blogs for people to compare the two on single cell data. Okay, so one last thing before I conclude the dimension reduction part is what we call independent component analysis. So for the independent component analysis, I think I am going to leave you to read the notes we post. So this is short as ICA. And this is different from UMAP and tSNE in that it is a linear dimension reduction. So I should have mentioned it earlier with Pca and Pnmf, but the reason I put it in the last is because we don't use it that often in biology. So that's why I put it in the last. This is linear dimension reduction. And many people confuse about it between the ICA and Pca. And I would say a simple way to distinguish the two is that Pca works the best for Gaussian-like data. So in other words, if you plot your data in every feature, you see that the n data points follow roughly a Gaussian distribution in every dimension. That's the case where Pca works the best. The reason is very simple. That is, if you think about a Gaussian distribution, it's specified only by 2 parameters. The mean, which is about the center, the location, and the variance or standard deviation, the same thing. They specify, so one is the square of the other. So variance is square of the standard deviation. They specify the spreadness. So basically, if you don't care about the center, that means you can shift your data as the Pca does. You just need to worry about the variance. So preserving the variance preserves the Gaussian shape. And ICA, on the other hand, it works for non-Gaussian data. So when you look at your data, it's clearly not Gaussian-like in every feature, then you might consider ICA. And ICA is, I think, its origin is from signal processing in engineering, in EE. So it's very widely used for speech analysis. Let me give you one example. Let's say we have several people, n people in the room, and we have placed n, yeah, it can be n, I'm not just using n. We'll use n microphones in the room for recording. So you can imagine that each microphone will record the voice from more than 1 people. And then given the n microphone recordings, we want to separate the signals so we can restore the original n people's voice. That's the typical application of ICA. So it's a signal decomposition step. And our goal, the meaning of independent, is that we want to find the n sources, like the n people's voices, as statistically independent as possible. So by statistically independent, that means, okay, for one source, whether its value being high or large, does not tell me whether the other sources of value at the same time being high or low. So that's what we mean statistically independent. It is similar to what we mean by orthogonal, but orthogonality or uncorrelatedness, that's directly equivalent to orthogonal. Having 0 correlation is only equivalent to independent for Gaussian data, not in general. So that's why here this independent is a more general way of saying orthogonality. You can think of it that way. And if we use the statistical way to do the ICA, we need some assumption on the distribution of the sources. We know it must be non-Gaussian, but we should specify it follows certain non-Gaussian distribution, and then we can use the maximum likelihood estimation to find the decomposition. That's one way, using the statistical or model-based way. We can also do ICA use the algorithm way, which means that we try to find an objective function and we try to optimize that objective function by minimizing it, for example, so that we can find the dimension reduction. So to do that, I can actually show you one interesting blog I found. So I think that can help you understand this better. So let me share that. And also I'm going to post a paper about the distinction between ICA and PCA for your future reading if you are interested. So first of all, let me share this. So this is the paper I'm going to post. So it gives the very detailed comparison between PCA and ICA. And like I said, I think for ICA it's very widely used for signal processing. So that's the example they gave here. While PCA is more used for, I would say, like gene expression data, like the data we used before, that kind of the matrix where you do not have a clear way to say, okay, my data are ordered in time. So you don't have many observations, in other words. So by observation, I need to clarify here, we have observations as cells and our features are genes. But in that case, we know our cells are not ordered just as in time. So it's not like a signal, but it's not like a continuous signal. And also, I think for our data, we tend to assume it's more like Gaussian-like. So that's why, in single-cell analysis, people prefer to do log transformation on counts to make every gene's expression across cells more Gaussian-like before they do PCA. They don't want to just work on the counts. And that's also why we said that's also aligned with the fact that PCA works better for Gaussian-like data. But ICA doesn't work for Gaussian-like data. That's something to keep in mind. If your data is Gaussian-like, then ICA will not give a unique solution. And this is the blog I'm going to share you. So yeah, I'm going to skip that motivation, but here, this is the mathematical formula. So you can think of this A as just your sources, and S is the, S as your sources, S for source, A is the mixing matrix. So you can think of like in the rows of X is the N people for N rows, and A will combine them into the microphone. So X is the observed signal. You observe X, you don't know A, you don't know S. You want to restore A and S from X. That's the ICA problem. And so you can see that, for example, we have 3 independent sources as the blue, green, and the orange. After we mix them into 3 microphones, here are the mixed signals. So we want to go from here, start from here, and try to find blue, red, and orange as our end result. Okay, so in other words, you see, given this formula, X equals A times S, and it's 3 by 3A, right? We have 3 sources, 3 microphones. If we can find the inverse of A, problem solved. We can just multiply inverse of A to the left of X. That's it. But how do we do it? Right, so how do we do it? And so there requires some condition for the ICA. So yeah, so maybe we can go through them one by one. The first precondition is that the mixed signal are linear combination of any number of source signals. That's what we said, linear dimension reduction, linear combination. The second precondition is that the sources are independent. That's the assumption we have for the algorithm. The third precondition is that independent components are non-Gaussian. That's what I said. Each source signal must be non-Gaussian. And here's what we mean. So if we think of this x-axis as source 1, y-axis as source 2, we plot them together. Each point is like one time point. What's the voice of person 1 or person 2? Then this is clearly not Gaussian. This is Gaussian. You know, Gaussian always have that, okay, dense around center, then getting less and less dense as you get away from the center. That's Gaussian-like data. But this uniform pattern is clearly not Gaussian-like. Just give you a very concrete example. And then if you do linear combination to get your mixed signals as a microphone, okay, so you can see the mixed signal for non-Gaussian-like data is like this. So then intuitively you say, okay, yeah, I just need some rotation and I need some stretching or contraction to restore the sources. Yes, indeed, we assume the sources, each source has standard deviation equal to 1. So we can address that identifiability issue. We do not have multiple possible ways. So we add that constraint standard deviation equal to 1. So it's given a mixed signal, you can just do rotation and stretching to restore the sources. But if the source is Gaussian, after you get a mixed signal, there are many possible ways to get the sources. In other words, you don't have a unique way to identify the sources from the mixed signal. That's just a property of Gaussian. It's invariant to rotation. That's the issue. So that's why you don't see a sharp angle as here and here for non-Gaussian data, but everywhere here, every direction here may be possible. So that's why for mixed signal, you cannot do ICA.
Dr. Lee?
But you can do, yeah.
So when we're saying it's non-Gaussian, I guess I'm a little confused because, you know, like there's, I guess, like if it's t-distributed, right? You know, and jointly t-distributed, then wouldn't have that have all of these same properties where it's kind of, it's rotationally invariant and you know, it's also, you know, clustered around the center. So I guess I'm a little confused about.
I would say multivariate T doesn't have that property as Gaussian. I would say that.
Okay.
So multivariate T is unlike multivariate Gaussian. So multivariate Gaussian is special in the case that whenever you do any linear combination of a multivariate Gaussian, let's say I collapse a multivariate Gaussian into a one-dimensional Gaussian in any way. So let's say I do this direction, I do this direction, I do this direction. And each direction will still give you a Gaussian. That's what we call, that's property of Gaussian. But I think multivariate T may not have that property.
Okay, got it.
Yeah.
But does it have to look uniform like this? I guess is the
I would say, so this is another thing I will share with you guys. So many people, when they do ICA, they try to have an object function related to the term called kurtosis. So kurtosis is actually regarding how skewed the distribution is, or I would think of it as how spread out the distribution is. So that's why I would say that the better, so the larger the kurtosis, the better the ICA will work. But I have to say, I'm also an outsider for this topic myself, because in my whole research experience, I never used ICA myself. So for me, this is something that I just read about, but I didn't really spend time to work on it. So I would say intuitively, if you look at this figure, you want to have some shape so that you can tell, okay, how I can restore a square. I think that's the intuition. Yeah, but if it's like well-rounded like this, then it's not possible. Yeah.
Thank you.
Thank you. Yeah. So yeah, and also this is another example. It shows that this is the mixed signal. It's like this, and after you restore the signal, it looks like this. But I would say, if you look at this gate, you can see also in each signal, it's actually quite uniform, right? Even though you may have some gaps in the middle, but it looks quite uniform. Yeah. And also, I would say to understand the algorithm, this is just one ICA algorithm. So what's below is just one type of ICA algorithm. It's not the only ICA. So this one is called a whitening. So what it actually does is simply this. So if you understand this figure, you understand it. So what that does is that it takes the covariance matrix of your data. So this is a P by P matrix. Okay, so if you do the centering, it's just X transpose X. For this covariance matrix, what you do is that you perform SVD, singular value decomposition. So once they get that middle part, this diagonal matrix, and the UMV. So I would think of it as the I think probably it actually meant the Eigen decomposition, because here this S is the eigen, the containing eigenvalues. Then what they did is that they tried to take the eigenvalue, take the square root to convert it to singular value, and then they take the inverse of it. So basically what you want to do is that you want to go from the current structure, transform it back to the original dimension, like here. And during the transformation, for example, okay, if in the new dimension, this part is really long, and then you want to know, okay, this corresponds to a very large eigenvalue, I want to contract it. So in the original dimension, I'm going to have the standard deviation equal to 1 here and equal to 1 here. So that eigenvalue is tell you how much you need to do the contraction. So the longer the new dimension, the current dimension is, the more contraction you need to do. So that's why you have the inverse here. And then with that, you just want to transform the matrix back. So it's still playing with the basis combination. So like U from here is actually it's kind of like your eigenvectors or the left singular vectors. So they are equivalent to each other. And what it actually means is how each dimension is a combined from the original dimension, like our loading. And you want to just rotate it back and also do the contraction. So yeah, I want to leave this for you. If, like I said, again, this is all about SVD and Eigen decomposition. And in the context of ICA, it's called whitening. Yeah, so I would say looking at this figure will give you the best illustration of ICA. So that's all I want to say about the ICA, and that concludes our dimension reduction part. So I think for today, probably I can just start a little about the next topic I want to talk about, although it will not be our major topic of this class, but still I think I will talk about it just because it's so widely used in our research. So the next topic I want to talk about is actually related to the first 2 homeworks in your next homework 3, which will be the last one in this quarter. So you'll have 3 weeks to work on it. And it's called the Generalized Linear Model, short as GLM, and Linear Mixed Effect Models, short as LMN. So although this is not a class for that topic, because if some of you know, or some of you are in my other class, I'm teaching a class specific for this topic, which is called 205. It's designed as an elective class for our statistics graduate students, including masters and PhDs. So in that class, we talk about these topics in great length. And here we do not have time to do it that way. So I just give you a glance about what's GLM and what is the linear mixed effect models. Okay, so let's first start with GLM. So generalized linear model. So clearly by the name, you can see there is a linear part and there is a generalization. So what does it mean? So roughly you can think of GLM is applicable to the cases where your responses are not real values continuous, are not continuous real values. So in other words, it's not a case where you think linear regression is reasonable. Think of linear regression. Okay, so if this is X, this is Y. So you know that your data points are like this, and you fit a line by linear regression. So given this line, you understand that the possible range of Y can actually be from negative infinity to positive infinity. So as far as the line goes, that's the linear regression. But your Y may not be the case. So for example, your YIs may be binary, 0, 1, or your YIs may be just non-negative counts like 0, 1, 2, and so on. So in that case, clearly you see this linear relationship is not satisfactory because your YI cannot really go down to negative infinity or go up to positive infinity. So that's the case where we need to generalize linear model. Linear model is not sufficient. So that's the motivation of GLM. And I think I will just, maybe if you have time, you can read what I wrote in the notes for this part, which I think is about the logistic regression, which is a special type of GLM. But of course, the linear regression itself is a special type of GLM. It's just that I try to motivate that to you to think, okay, linear regression itself is not sufficient. So we need to generalize it. And that gave us GLM. So in the next class, I'm going to continue this part. And also I'll invite a PhD student of mine to give you a very concrete example about how he used GLM in a collaboration project with a biology lab at UCLA. So I think that can give you some sense about how you may consider GLM in your research. And also, if you remember, we talked about DE-SIG2. Yeah, that DE-SIG2 example, we take a brief look at what it does. And that paper actually uses GLM to solve the problem of differential gene expression. So what it does is it tries to fit a generalized linear model for each gene. And it considers, so 1 GLM per gene. And it considers the condition as a predictor or covariate. So these are terminologies used in the field, and then check if the conditions coefficient is 0. So that's the idea. So then it does a statistical test. So you can use this GLM as a general way of comparing two conditions. So you can think of, if you are familiar with like a two-sample t-test we talked about in the beginning, then that can also be considered as a special case of a linear regression. You could do linear regression with a condition as a predictor, check whether its coefficient is equal to 0. So that is actually roughly equivalent to doing a two-sample t-test with some slight differences. And also for GLM, you can think of it can handle data that are not continuous real values, but maybe binary counts. So for those cases, GLM is a very useful tool. Okay, so it's a tool for doing association study. Let me put it that way. All right, so let's stop here and I'll see you on Thursday.